In today's session, we will study connective tissue. As we know, there are four basic tissues in our body. First is epithelium, which forms outer covering and inner lining of the body organs. Second is connective tissue, which gives support and it forms structural framework for various organs of the body. The third basic tissue is muscle. So it has contractile unit which helps in locomotion, and the fourth basic unit is nervous system, neuron. So it helps in transmission of impulses. So there are four basic tissues of which we will study connective tissue. So the connective tissue is one of a basic tissue which gives structural and metabolic support to the organs of the body. It has certain general features. So they are: the connective tissue is composed of cells. These cells are of different types. And the second component is matrix, which is made up of fibers and ground substance. So the fibers can be collagen fibers, elastic fibers, or reticular fibers. Connective tissue develops from mesoderm, with few exceptions like neuroglia. Now we know in central nervous system we have neurons and the neuroglial cells. So neuroglial cells form support. They are supportive cells and they include astrocytes, oligodendrocytes and microglial cells. So astrocytes and oligodendrocytes are neuroectodermal in origin, whereas microglial cells are mesodermal in origin. Then the pigment cells present in skin, iris, retina are ectodermal, and the reticular cells of thymus are endodermal. Thus, we can conclude that connective tissue mainly develops from mesoderm, with few exceptions like neuroglial cells, pigment cells, and reticular cells of thymus. The cells present in connective tissue are of different types, and between these living cells is the non-living intercellular substance. So the functions of connective tissue are: it provides mechanical as well as structural support to the organs. The intercellular spaces are filled with the connective tissue, so it acts as a packing material. Then the organs are covered by capsules. They provide shape to the organ. Again, through the capsule goes the trabeculae and septa. They divide the parenchyma of the organ into lobes and lobules. Then if we take example of blood, it is also a connective tissue which acts as a medium for exchange of nutrients and waste products. Then the adipose tissue stores fats in it. So it stores energy and there is thermal insulation. The loose connective tissue stores water and electrolyte. The fibroblast cells of connective tissue can produce matrix. They can synthesize fibers, all the three varieties of fibers. So they help in repair. And the lymphatic system, the B lymphocytes can produce antibodies. Then mononuclear phagocytic system can engulf microorganism. So there is defensive function also. And there are some trophic and morphogenetic roles of connective tissue which help in growth and differentiation of the tissues. In connective tissue, there are four types of cells. So those cells which are metabolically active and those cells which can divide mitotically, such active cells are known as blast cells. Those cells which are metabolically inactive and they are in resting phase and they do not divide mitotically are known as site cells. Then few cells have property of phagocytosis. So they are phagocytic cells or scavenger cells. And in case of bone, the bone absorbing cells are known as osteoclast. And developmentally, there are two types of cell lines. Embryonic cells are, or the prenatal cells are resident, means they are fixed at one place. These native cells mainly originate in spleen, bone marrow and liver. Second variety is postnatal or immigrant. So they can transfer, or these are also known as wandering cells. So they mainly originate from undifferentiated stem cells in the bone marrow. Connective tissue is broadly classified into two groups: connective tissue proper and specialized variety of connective tissue. So in specialized variety there is cartilage, bone, blood and adipose tissue, which we will study in detail in later sessions. Now the connective tissue proper is further divided into embryonic connective tissue and adult connective tissue. Embryonic connective tissue again has mesenchymal type and mucoid type of connective tissue, whereas adult connective tissue is divided into loose and dense connective tissue. Depending on the arrangement of collagen fibers, the dense connective tissue is again divided into dense irregular and dense regular connective tissue. Three main components of connective tissue are cells, fibers, and intercellular ground substance. So the cells can be fixed or native or resident cells, and the second is immigrant or wandering cells. So the fixed cells present in connective tissue are fibroblast, adipocytes, macrophages, mast cell, pigment cell and adult stem cells. And the wandering cells mainly present in the blood are lymphocytes, plasma cells, neutrophils, eosinophils, basophils and monocytes. Then the second component is fibers. So they are of three variety: collagen, elastic and reticular fibers. The intercellular ground substance is mainly formed by either proteoglycans or glycoproteins or glycosaminoglycans. So the three components of the connective tissue are cells, fibers and intercellular ground substance. Fibers and intercellular ground substance together are known as matrix. This slide is of loose areolar tissue. In this slide, we can see all the components of connective tissue. So these pink bundles are the collagen fibers. Okay, so these are the branching bundles of collagen fibers, and they stain pink with eosinophil. So these are eosinophilic and they stain pink. Then the second component is the elastic fibers. So you can see these thin black thread-like fibers. These are elastic fibers. Then the third type of fibers are reticular fibers. They cannot be seen by routine H&E staining, and we need silver impregnation to see reticular fibers. Then the second component is cells. So we can see different types of cells in the connective tissue. So these cells are plasma cells. They have cartwheel appearance of a nucleus. So these are plasma cell. Here these are the macrophages. Then these are mast cells with secretory granules in them. Then we can see small as well as large lymphocytes in it. Whereas the ground substance, it is pale stained. Thus all the three components of connective tissue, cells, then the fibers and the ground substance can be seen here. Now these cells are the fibroblast cells. There are three types of fibers in the connective tissue. First type is collagen fibers. They are present in all the connective tissues. Then the reticular fibers are mainly seen in the basement membrane, lymph node and liver. And the third variety of fibers are elastic fibers. They are present in the blood vessels, mainly in tunica media. Then the lungs have elastic recoil. So it is due to presence of elastic fibers in it. Then ligamentum nuchae, ligamentum flava, they also have elastic fibers in them. The collagen fibers are strong, flexible, inelastic, and they have tensile strength. And they are synthesized by fibroblast cells. Routinely in H&E staining, they take pink color as they are eosinophilic. With the help of methylene blue dye, then Mallory stain, then Mesenchymal, in all these dyes they'll stain blue. Under light microscopy, the collagen fibers appear as bundles. The individual fiber can means the individual fibers do not branch, but the bundles, the wavy bundles which appear pink in HNA staining, they may branch. And there are 29 types of collagen fibers, and this classification is based on the sequence or chain of alpha chains. Based on the sequence of alpha chains in the amino acid, the topocollagen or the collagen fibers are broadly classified into 5 types, and we will see the location of each. So type 1 collagen fibers are most common variety of fibers, and mostly they are found in bones, tendons, ligaments, dentin of the teeth, dermis. And if there is deficiency of type 1 collagen fibers, it leads to osteogenesis imperfecta. The type 2 collagen fibers are located in hyaline and elastic cartilage. They are also seen in vitreous of the eye and nucleus pulposus and notochord. So if there is deficiency of type 2 fibers, it leads to kinase dysplasia. Type 3 fibers are also known as reticular variety of collagen fibers. They are located in loose connective tissue of a liver, spleen, lungs, kidney and uterus. And if there is deficiency of type 3 reticular fibers, it leads to Ehlers-Danlos syndrome. Type 4 fibers are present in the glomeruli of kidney, basal lamina of epithelium, and they form the filtration barrier of a kidney. And if there is deficiency of type 4 fibers, it leads to Alport syndrome. Type 5 fibers are seen in placental tissue. This slide we will see synthesis of collagen. So first step is translation of mRNA present in the nucleus by ribosomes. And after translation of mRNA, there is synthesis of pro-alpha chains. And these will form and stabilize procollagen. So it is a triple helix molecule, and the procollagen is then transported to the Golgi apparatus, and packing of procollagen occurs in the Golgi apparatus, and it will form the secretory vesicles, and these all steps occur in the cell, okay, in the nucleus and then in the intracellular uh, space. And after the formation of secretory vesicles, these are excreted by the process of exocytosis out of the cells. So exocytosis of procollagen by secretory vesicles occur, and now these are present in the extracellular space. And first there is conversion of procollagen to tropocollagen by enzyme procollagen peptidase. Then the polymerization occurs, and finally the collagen fibers are formed and their bundles are seen. Aggregation of collagen fibers will form the bundles. Collagen fiber provides tensile strength. Collagen present in the skin helps in stretching of the skin. So if we see the skin of young person, it is glossy and it can be stretched, but as the age advances, the production of collagen fibers is decreased, and in old age thus wrinkles appear on the skin. It is due to decreased production of collagen and elastic fibers. Then the collagen fibers are present in the deep fascia, like fascia lata of the thigh. Then the deep fascia forms a sleeve around calf muscles of the lower limb. Now we know the calf muscles are gastrocnemius and soleus. So these are surrounded by a tough sleeve of deep fascia, and the soleus has got venous blood in it, and when it contracts it helps in the venous return against gravity of lower limb. So this is also helped, or it is assisted by the deep fascia which surrounds it, as it provides a tough unyielding surface. Then the ligaments also have collagen fibers in them, and they help in holding the bones firmly, and the cartilage and bone has recoil property due to presence of collagen fibers in them. If we take the example of eyeball, sclera and vitreous body, both have collagen fibers in them, and they help in maintaining the shape of eyeball. Then the muscles contract, and the collagen present in the tendon also gets stretched, and it helps in locomotion. The reticular fibers are arranged in the form of meshwork or network, and they are mainly composed of type 3 collagen fibers. Reticular fibers are located in liver, intestine, lymphatic tissue, glands, nerves, bone marrow, and muscles. So these reticular fibers mainly form a structural framework, and supportive framework is formed in which the parenchyma or the tissue is laid down for an organ. And the reticulocytes are mainly produced or synthesized by reticulocytes or the fibroblast cells, and they do not take routine H&E staining. For seeing the reticular fibers under microscope, we need silver impregnation, and they appear black in color. They are argyrophilic, and they are also pass positive. Its main function is to form supportive framework for many organs and the glands, and in the basal lamina of the basement membrane, they support the epithelial tissue. We know that the epithelial tissues resting on the basement membrane forms hemidesmosome, that is junctional complex, and thus the basal lamina, the reticular cells in the basal lamina will support the epithelial tissues. Third variety of fibers are elastic fibers. So the elastic fibers are very fine hair-like or thread-like fibers, and they mainly form again plexus or anastomosis, and they branch. The individual elastic fiber can also branch, and they have property of stretching and recoil. And valine is the amino acid which is highest content of elastic fibers. Elastic fibers are synthesized by fibroblast, and sometimes smooth muscles of the blood vessels will also form elastic fibers. They are located in skin, blood vessels, ligamentum nuchae, ligamentum flava, elastic ligaments of vocal folds, and lungs. Then on electron microscopy, there is a central core of amorphous material surrounded by outer layer of fibrils. The elastin is the central core. So the central core is surrounded by fibrillin 1, and it stains dark brown with orsin, and it stains blue-black with the Wiegand's resorcin fuchsin stain. Weirhoff stain is also used. So these are the special stains used to visualize elastic fibers under microscope, and its deficiency will lead to Marfan syndrome, that is abnormal fibrillin gene located in 15th chromosome. Elastic fibers are present in large arteries and large veins. So the great vessels of the heart have more amount of elastic fibers in them. So if we take example of arteries, the aorta, pulmonary arteries, then the carotid and subclavian artery are having elastic fibers in mainly in the tunica media of their wall, and they help to pump blood away from the heart. And in large veins like superior and inferior vena cava, the elastic recoil of the vessels will help to pump blood in the right atrium. Then in medium-sized arteries, we can visualize internal and external elastic lamina under microscope. So these laminas will help to keep the lumen open. So the lumen is kept patent during blood flow. In the pinna, elastic cartilage helps to collect sound waves, and in epiglottis, the epiglottis acts as a flap cover, so it covers the laryngeal inlet during deglutition. In the figure given below, we can see all the three types of fibers: collagen, elastic, and reticular fibers. And in this table, we can make out the difference between the three types of fibers. So mainly the collagen fibers are located in tendon, ligament, and organ capsules. Elastic fibers are located in large vessel, lungs, and ligamentum nuchae, and reticular fibers are present in the basal lamina, dentin, pulp of the teeth, liver, spleen, and lymph nodes. And we use special stains to visualize these fibers. And if we see its properties, the collagen fibers have high tensile strength, the elastic fibers are highly elastic, and the reticular fibers, they have low amount of elasticity as well as tensile strength. So far we have seen the three types of fibers. Now we will see the types of intercellular substance, that is the substance which fills the spaces between the cells. So it can be fluid, for example lymph, then cerebrospinal fluid, aqueous humor in the eyes, then it can be viscous like blood plasma, then the soil, it can be oily, for example synovial fluid present in the joint cavities, and amorphous ground substances most abundant, and this can be glycosaminoglycans. So the glycosaminoglycans have the property of trapping the water molecules, which gives it gel-like appearance or consistency. And then it can be firm like in cartilage, or it can be hard like in bones. In bones, the intercellular substance is hard because there is impregnation of inorganic salts like calcium and phosphorus. And in cartilages, the intercellular substance is firm because there is sulf means the glycosaminoglycans get sulfated in cartilage, and that makes it more firm and flexible. The ground substance when stained, it is positive with periodic acid scheme. So the composition of extracellular matrix is, the first component is glycosaminoglycans. So as I told you earlier, it has the ability to trap water, so it gives it gel-like appearance. And there are five major classes of extracellular matrix. So hyaluronic acid is non-sulfated, and it is mainly seen in cartilage, umbilical cord, and vitreous body. Then chondroitin sulfate is present in cartilage, bone, skin, and cornea. Dermatin sulfate is present in skin, blood vessel, heart walls, and lungs. Keratin sulfate in cornea cartilage. And heparin sulfate is present in the aorta, liver, and lungs, and also in the mast cell granules. So the first composition and most common composition of extracellular matrix is glycosaminoglycans, and we have seen five major classes and their location. Other compositions of extracellular matrix are proteoglycans. They have aggregans and syndicans. So the core protein here provides attachment to many glycosaminoglycans, and it gives a bottle brush like appearance. And when there is degradation of lysosomal cells and lysosomal enzymes, it leads to deficiency of lysosomal enzyme, and the condition is known as lysosomal storage disorder. Third composition is glycoproteins. They are mainly for stabilizing the extracellular matrix, and they are located in laminin. So laminin is the glycoprotein present in the basal lamina. Then fibronectin provides attachment of fibroblast to collagen fibers. Osteonectin provides attachment of bone cells to the collagen fibers. Chondronectin provides attachment of chondroblast to the collagen fibers. Then we also have intactin and tenascin. So these are all glycoproteins. The functions of ground substance are: it provides morphological and structural framework. Then it binds the cells present in the intercellular spaces. Then it acts as a mechanical barrier, for example, glomerulus of a kidney. Then if we take example of basement membrane, it helps in diffusion of metabolites. Then there is storage of water, carbohydrate molecules by hyaluronic acid. Coming to the cells of connective tissue, so there are two varieties again: fixed, native or resident cells like fibroblast, adipose cells, macrophages, mast cell and parasites. And the wandering cells or immigrant cells are lymphocytes, then the plasma cells, neutrophils, eosinophils, basophils and monocytes. So we can visualize both the varieties of cell in the diagram. The first variety of cell is mesenchymal cell. So mesenchymal cells are known as precursors because they can give origin to all types of cells present in the connective tissue, and they are arranged along the blood vessels, and they are small stellate cells, and they have tapering ends. As we can see in the figure, they have slender processes, and these slender processes join with one another and they form a fine network. Fibroblast, these are derived from undifferentiated mesenchymal cells, and these are metabolically active. They can mitotically divide, and they can produce and maintain extracellular component, means it can synthesize fibers as well as ground substance. And they have abundant amount of cytoplasm, oval nucleus and branched cytoplasmic processes as shown in first figure. And on H&E staining, they are visible as thin, pale, flat processes are visualized, and we can also visualize the violet nuclei in them. And these are more active during wound healing as they help in the healing process. They are active. Then the fibrocyte, in second diagram we can see the fibrocytes in resting phase. They appear spindle shaped, means they are fusiform. There is bulging central part and the ends are tapering. And it is inactive and mature, and these are smaller cells with heterochromatic nucleus. And the function of fibroblast is mainly tropocollagen proteins are synthesized by it, which helps in the formation of collagen fibers. Then they produce proteoglycans, and this helps in synthesis of reticulin and elastic fibers. Then they are very active in the wound healing process, and with the help of myofibroblast there is wound contraction. And they also act as stem cells, as these are undifferentiated mesenchymal cells. They can give rise to the fibers as well as the ground substance. The myofibroblast is a variant of fibroblast, and these are again elongated spindle shaped, and they mainly contain actin and myosin filament in them, which offers contractility. So during wound healing also they cause wound contraction, and they share property of both smooth muscle cells and the fibroblast. So the second type of cell is pericytes. So as you can see in the diagram is of a capillary. We can see the endothelial cells of the continuous capillaries, and in vicinity, we can also see the parasites. So these parasites have long euchromatic nucleus and several cytoplasmic processes are there that help in clasping the capillaries. So the parasites are surrounding the endothelium of capillaries and venules in the basal lamina of endothelium. So the next variety of cells present in connective tissue is adipocyte or lipocyte. These are also known as fat storing cells. So they store energy and they help in thermal insulation. So if we see in the diagram, these adipocytes are spherical in shape, and when many cells are there, they are closely opposed to each other, and thus it gives polygonal shape. So these cells have thin rim of cytoplasm and eccentric nucleus, and there is a large central lobule of fat. So this central lobule of fat will shift the cytoplasm to the periphery. So there is only a thin rim of cytoplasm, and also the nucleus is shifted to the periphery. That's why nucleus is also eccentric, as we can see in the diagram. And in routine H&E staining, it gives signet ring like appearance because the central lobule of fat is washed off by xylem. So the cell appears empty in the central part. We can use special stains to visualize the adipocytes, like there is Sudan black B stain, then the cells also stain with Sudan 3, they take orange color, and adipocytes can be unilocular or multilocular. So unilocular adipocytes are also known as white or yellow adipocytes. So we commonly observe it under microscope, and that is unilocular, and these are present in the adult, and their function is insulation and shock absorption. They also synthesize and store lipids. They are mainly located in surrounding the vital organs, so it will protect and support the vital organs. Then it forms mammary pad of fat. They are surrounding the orbit, retroperitoneal space, and it is also present in the greater omentum. And multilocular variety of adipocytes is also known as brown adipose tissue, and it is mainly seen in newborn babies, and its function is thermal regulation. So in adults it is only present in small amount in the back and neck, and on also in the scapular region, and its function is to prevent excessive heat loss and to promote thermoregulation in. The macrophages are also known as histiocytes or plasmatocytes, and if we see in the figure, they are either fusiform or they can be spheroidal or stellate shaped, and they stain darkly with they have very small indented nucleus at one end, and the cytoplasm has variety of granules, and it can be stained with vital stains like lithium carmine. And here we can see when we stain it with the vital stains, with the help of phagocytosis, the dye will be accumulated in the cytoplasm, and the main function of macrophages is phagocytosis of microorganisms. They also phagocytose damaged tissue and inorganic particles, and they act as antigen presenting cells. Then there are two types of macrophages, fixed and wandering. So wandering cells are active, and when they are active they become free, they are ovoid, or and they have amoeboid movement, and the cytoplasm has numerous lysosomes in them. Fixed macrophages are inactive, they are in resting state, they are fixed, and they have irregular outline, and they show philopodia and hyperchromatic nuclei. So these are the two differences, histological differences between fixed and wandering macrophages. Then there is a mononuclear phagocytic system, in which the macrophages are present at different sites, and the Langerhans cells in the epidermis part may form a giant multinucleated cell, and the monocytes are also present in blood. Then the Kupffer cells in liver, microglial cells in the central nervous system, alveolar cells of the lung, Hofbauer cells in placenta. These are all examples of mononuclear phagocytic system. And the reticular cells in the lymph nodes also belong to the same group. Mast cells are also known as histaminocytes or mastocytes. So as we can see in the diagram, these are ovoid cells, large ovoid cells, and they have centrally placed nucleus, and the cytoplasm is basophilic. It has granules which contain heparin and histamine in them. And the mast cells are located close to blood vessels, serous membranes like pleura and peritoneum. And when we stain it with toluidine blue or Alcian blue, the granules are seen blue in color with methylene blue also, and they show metachromasia. Metachromasia means the granules look purple to red with toluidine blue, and they have immunoglobulin E present on the cell surface. And as we have earlier discussed, that it has granules which contain heparin and histamine in them. So the cells release histamine that has allergic reaction. Heparin is an important anticoagulant. Then the serotonin is a vasoconstrictor, also secretes serine protease and leukotriene. Then the brain and spinal cord is devoid of mast cell, and thus brain is protected from the harmful effect of edema and other allergic reactions due to mast cells. Then the next variety of cell is lymphocytes. So these are deeply stained heterochromatic nuclei is there, and they have thin rim of cytoplasm. So they can be large or small in shape, and they are mainly involved in immune response. So there are T lymphocytes and B lymphocytes, and also we have natural killer cells. The plasma cells are also known as plasmatocytes, and these cells are derived from B lymphocytes. And again, as you can see in the diagram, they are ovoid in shape. They have abundant cytoplasm, and the cytoplasm has rough endoplasmic reticulum in it. The identifying feature of plasma cell is presence of cartwheel appearance of the nucleus. So the chromatin in the nucleus is arranged in radial pattern, which gives it cartwheel appearance, and the nucleus is eccentric. Then they have strong affinity for basic dyes, and Russell bodies are the large immunoglobulin containing inclusions, and these are known as Maillot cells. So the Russell bodies are also positive for periodic acid skip stains, and the plasma cells produce antibodies. They are located in lamina propria of respiratory and gastrointestinal tract, and there is a malignant tumor known as myeloma, in which there is proliferation of particular clone of plasma cells in the bone marrow. So the proliferation of plasma cells in bone marrow is known as myeloma, and it is malignant in nature. The pigment cells are stellate cells, and they have branching processes. The cytoplasm of pigment cells show dark brown pigments in them, and they can be visualized with the help of DOPA. So the melanocytes are the cells which have round nucleus and long processes, and they can produce the melanin pigment. So the cells containing melanin granules in membrane bound organelles are known as melanosomes. And there is other means a different entity known as melanophore. It is different from melanocyte. Melanocyte are melanin producing cells, but there are some cells which do not synthesize melanin by their own, but they can engulf the melanin pigment. So when it appears, means when they engulf the melanin pigment, they appear brownish black, and these are known as eumelanin, and they appear reddish yellow when and at that time they are known as pheomelanin. Now if there is complete absence of melanocytes or melanin, if there is absence of melanin, generalized absence, it is known as albinism. At foci, if such melanocytes are deficient, it leads to vitiligo, and when the melanocytes are absent, it makes the skin prone to malignancy because of the harmful effect of ultraviolet rays, and there can be malignant melanoma also. So the main function of melanocyte is to give complexion to the skin, and the pigment cells in the retina, in the iris, they protect the eye from harmful effect of UV rays. So here the applied significance, the terms you should know is albinism, where there is generalized absence of melanocytes and the melanin pigment is absent in such individuals. When focal absence is there, it is known as vitiligo, and such individual in whom the melanocyte or the melanin pigment is deficient, these are more prone, or these are more susceptible to the malignancies of skin due to hazards of ultraviolet rays. The white blood cells includes neutrophils, basophils, eosinophils and lymphocytes, and we have seen monocytes also in earlier diagrams. You remember the classification of connective tissue as discussed in earlier slides. We have seen the connective tissue is classified broadly into two groups: connective tissue proper and specialized variety. Connective tissue proper is further divided into embryonic variety and adult variety. Embryonic is divided into mesenchymal variety and mucoid variety. So the mesoderm gives rise to embryonic connective tissue, and there are two types: mesenchymal and mucoid type of connective tissue. So in the first diagram we can see mesenchymal connective tissue. It is found in embryo, and the spaces between the cells are occupied by ground substance, reticular cells and collagen fibers, and the adjacent cells are connected to one another by gap junction. It is mainly located in the pulp of teeth, vitreous of the eye and nucleus pulposus. So the mesenchymal variety of embryonic tissue is located in nucleus pulposus, vitreous of eye and pulp of teeth. Second variety is mucoid or mucus variety of connective tissue. It is exclusively seen in Walton's jelly of umbilical cord. So the umbilical cord has two components. There are cells and Wharton's jelly. Then we can see in the diagram there are two, right and left umbilical artery, and the left umbilical vein is there, and in between we can see Wharton's jelly which has cells in it. So the cells are spindle shaped and they have long cytoplasmic process, and the Wharton's jelly mainly consists of hyaluronic acid. Built variety of connective tissue is divided into loose or connective tissue or areolar connective tissue and into dense connective tissue. So there are two subtypes of adult variety: loose tissue and dense tissue. So the loose areolar tissue, areola means what? Empty spaces. So as many empty spaces are there and as the connective tissue is loosely arranged, it is known as loose areolar tissue, and it has three components. Cells are there, we can see all the different types of cells of connective tissue in it. Then the fibers, all types of fibers, collagen, elastic and reticular fibers are there, and the extracellular matrix has more amount of areola or empty spaces in them. The loose connective tissue is mainly seen in lamina propria. Then now where is this lamina propria? If we consider any organ, for example in gastrointestinal system, we have four layers: mucosa, submucosa, muscular layer and serosal layer. So in the mucosa we have lining epithelium, and beneath that is the lamina propria. So here we have loose areolar tissue. Then during dissection also we have seen, we first incise the skin, and beneath the skin is the superficial fascia, so it is laden with fibrofatty tissue. It is an example of loose connective tissue. Then surrounding the blood vessels and viscera also there is loose connective tissue. And now if we take the example of veins, the veins are surrounded by such loose connective tissue. So whenever there is increased venous return, the veins get space for expansion. Main function of loose connective tissue is to provide support. It allows movement as there is space in between, so there is um, a space for movement and also space for expansion of viscera and blood vessels. So the loose areolar tissue is located in the subcutaneous part of eyelids, scrotum, penis, submucous coat of gastrointestinal tract, and so on. And if we see histologically the structure of loose areolar tissue, the intercellular ground substance is soft, jelly-like. It is mainly made of mucopolysaccharides or glycosaminoglycans, in which all the three types of fibers, collagen, elastic and reticular fibers are there. And all varieties of cells, mainly the fibroblast, fibrocyte, mast cell, macrophages are present. And on routine H&E staining, the ground substance appears faint. Then the collagen fibers take pink color with the eosin, and the cytoplasm is pink, and the nucleus of the cells appear blue in color. We cannot differentiate different types of cells under low power, but we can see their nuclei. The violet color nuclei of different cells are visualized under low power. And the main function of loose areolar tissue is they form packing material between muscles and vessels, and they provide elasticity as well as tensile strength, as they have elastic fibers and collagen fibers in them. Then the extracellular tissue fluid provides exchange medium between capillary bed and cells in the connective tissue. Again, if we recollect the classification of connective tissue, the adult variety of connective tissue is classified into loose areolar and dense type of connective tissue. So the dense connective tissue has more amount of fibers and less amount of cell and matrix. That's why it appears dense. And as it has more amount of fibers, it provides strength and protection. And again, depending on the arrangement of the fibers, dense connective tissue is of two subtypes: dense irregular and dense regular. Dense irregular connective tissue, as you can see in the diagram, has got intercellular matrix which has more amount of fibers, and these fibers, elastic and collagen fibers, are arranged irregularly. There are irregular network of such fibers, and the ground substance is less in amount. Cells are mainly fibroblast, and dense irregular connective tissue is located in the dermis of the skin, in the dura mater. Epineurium is the supportive framework of the nerve fibers. Then periosteum is the outer covering of the bone. Perichondrium, outer covering of the cartilages. Then dense irregular connective tissue is also seen in sclera, tunica, adventitia of large vessels. One variety of dense connective tissue is dense regular connective tissue. So it has more amount of type 1 collagen fibers, and the bundles of the collagen fibers show wavy appearance under H&E staining. And the dense regular tissue is mainly found in ligamentum flava, then the vocal cords, And you should remember the example of tendon. We have this slide of tendon for practical also. Then the dense regular tissue is also present in ligaments, aponeurosis. And if we see structurally, the tendon in tendon, the collagen fibers are arranged in bundles, and in between these bundles, there are rows of flattened fibroblasts, and there is minimum amount of intercellular substance. The tendon is how the tendon is formed. So if we see the muscle, now where the tendon is located, if we see the muscle, it has a central part which is known as belly. So the belly is fusiform in shape, and on either side it has tough fibrous tendon that is at the periphery. So central is belly, and at either end of this there is tendon over the muscle. So the muscle has got individual muscle fibers covered by endomysium, and these muscle fibers will form bundles or fasciculi. They are covered by perimysium, and the entire muscle is covered by epimysium. So these endomysium, perimysium and epimysium are the connective tissues which are forming supportive framework for the muscle. So all these three coverings will extend peripherally and they will form the tendon. So collagen fibers in the tendon enters into the compact part of the bone, and it remains unossified, and it is known as Sharpie's fibers. So these are anchoring fibers at the end, and the anchoring fibers get ossified, whereas the Sharpie's fibers remain unossified. So this is how the tendon is formed. The main function of tendon is when the muscle contracts, the tendon becomes tense and it pulls the bone by its tensile strength, and there is locomotion. And in slides we have longitudinal section as well as transverse section of the tendon. So in longitudinal section we can see the bundles of collagen fibers which stain pink. In between we can see fibroblast, fibrocytes. They are flattened, and we can see their violet colored nuclei in rows. And transverse section mainly shows the supportive framework of connective tissue. So uh, here the bundles are surrounded by loose connective tissue. Again we can see fibroblast and fibrocytes in between the collagen bundles. In this slide, we have two diagrams of tendon, longitudinal section and transverse section. So if we see the composition of tendon, it has type 1 collagen fibers, more amount of type 1 collagen fibers. Then the cells are fibroblast and fibrocytes. These are also known as tendinocytes. And we can also see the covering uh, framework or connective tissue covering the tendon. So endotendinium covers the bundle of collagen fibers, and epitendinium covers the entire tendon. And these connective tissue provide passage to the blood vessels. So in the second slide we can see the transverse section in which the fibers, collagen fibers, and those blue dots are the nuclei of fibroblast and fibrocytes, and in between we can see the connective tissue, supporting connective tissue which has blood vessels in them. So adipose tissue is specialized variety of connective tissue, and it is highly vascular, and there are two varieties of adipose tissue: unilocular or yellow adipose tissue, which is commonly seen even in adults, and here the nucleus is flat towards the periphery. There is thin rim of cytoplasm, and there is typical signet ring appearance of the adipocyte, and it is mainly it is located in the superficial fascia and yellow bone marrow, and its function is protection. The second variety of adipose tissue is the brown adipose tissue. It is multilocular, and it is mainly seen in newborns, and its function is regulation, that is regulation of temperature. It prevents heat loss, and it also acts as a reservoir of energy. And the adipose tissue maintains the body contour, it supports and protects the organs. It also acts as an insulin. So this is the diagram of yellow variety of adipose tissue, which is highly vascular, and we can see the HNE staining appearance as the fat droplets are washed off during staining process. These appear empty with thin rim of cytoplasm and peripheral flat nucleus. Second variety of adipose tissue is brown adipose tissue. It is multilocular, the cells are smaller, and it is mainly found in newborns, and it is meant for thermal regulation. Coming to the clinical aspects of connective tissue, there is a condition known as scleroderma. It is autoimmune disorder of connective tissue, and there is slowly progressive rheumatic disease where the thickness of the skin increases due to deposition of fibrous tissue. And the second condition is Dupuytren's contraction. So we know in the palm there is, means after we remove the skin, there is palmar aponeurosis, and the palmar aponeurosis, again in that there is contraction of fibrous tissue, and this disease results in flexion deformity of fingers, and there is flexion at interphalangeal joint, especially in the ring finger and little finger due to contraction of fibrous tissue.